Our second speaker is Carlo Colantuoni, who is an assistant professor of neurology and neurosciences in Johns Hopkins School of Medicine. He got his undergraduate degree from Princeton University and went on to do his PhD in Johns Hopkins School of Medicine, which he graduated in 2002. And today, the work he will be presenting is a product of a long collaborative effort between him and many others. He's leading the computational elements, and cell work is done from Yale by Soo Kim, and they are focusing on elucidating the origins of diversity in the emergence of the neural lineages from pluripotency, as a way to explore individual human cell biology, health, and diseases. So yeah, Carlo.
Thank you. So as she mentioned, Soo-Ki Kim has conducted the cell work, but this all started as a collaboration in the brain development group of Ron Mackay here in Baltimore. We have since moved on to different institutes, but maintained our collaborative effort on really investigating what we feel is key aspects of differences in human pluripotent stem cells, and really on the road to understanding differences between individual humans, as we see here. So this is where things start. These are pluripotent stem cells grown in a monolayer. Each of the different panels is a different line here. And this is after 4 days of NSB treatment, which is NOG-NSB. SB is a small molecule, and this is dual SMAD inhibition, which which induces the neural lineage from pluripotent stem cells. And as you can see, even before we look at what genes are on and off here, that there is a very big difference in the efficiency of the induction of the neural lineage from pluripotence across these different lines. So in green, we have NANOG, the pluripotency marker, and in red, we have SOX21, showing the cells and the region of these two-dimensional cell clusters, cell colonies that are inducing the neural lineage. And I'll leave it at this image for now, but Soo-Ki measured this attribute, the differential emergence of the neural lineage across these lines in many different ways and in many different experiments. And it is a stable trait or a stable phenotype of the different lines, that they are less or more reactive to the dual SMAD inhibition and the neural induction. So we really want to get into exactly what's in this first simple panel is how and why, or I guess more how, is one cell line different than another in this early emergence of neural lineage. So the data set that's going to be sort of at the core of most of my talk is an RNA sequencing data set, bulk RNA-seq, using these 6 lines that are differentially emerging from pluripotency. And we have, with the 6 lines, we have 3 different conditions. In black, self-renewal, so simply pluripotency, NSB, the dual SMAD inhibition, and then BMP4 inducing mesendodermal lineages in blue. So we have days 2, 4, and 6 for each of these 3 conditions in all 6 lines. This is a PCA plot of that experiment where we can see in black, the self-renewal is this cycle where all the cells are kind of in this whirlpool of pluripotency. As you inhibit SMAD signaling and induce the neural lineage, they kind of slingshot out of that space. You can see the different lines come out at different speeds as 2, 4, and 6 days emerge. This SAO1 line is the quickest in the neural lineage. And in the BMP condition, you can see immediately at day 2, there's a huge shift. So in that first 48 hours, a lot of things change with BMP, as you might expect, being the sort of in vitro analog of the beginning of gastrulation. And then here also, although they all are in a similar space here, they move through it with different efficiencies. For instance, the I4 line here highlighted in green, by day 2, it's not only all the way back here, but all the way up here. So clearly all 6 cell lines move through some neuroectodermal and mesendodermal lineages, but there's differential efficiency or speed which they move through there. So now we're going to really look at this RNA sequencing experiment and get into more granular resolution. So we used a non-negative matrix factorization And if you're interested, we can talk offline about what that is mathematically. But basically, it's a different way to break down the signals in any high dimensional matrix to a low dimensional format. And so we looked at 22 patterns that we could find in that RNA sequencing experiment. And here's 2 of those where we are visualizing on the x-axis the 2, 4, and 6 days. And the different colors are the BMP, dual SMAD, or self-renewal time course. So pattern 7 of 22 turned out to be a pluripotency pattern, high in the black cells, or in the black lines here, which are the pluripotent cells. When you stimulate with BMP, it's shut down almost, entirely before 2 days shows up, so it's flat here. And when we do the dual SMAD inhibition to induce the neural lineage, it comes down sort of slowly, but kind of intermediate there. And here's an example of a BMP-induced pattern that goes up, and again, there's differences between the lines here, but it goes up over time. And to understand really what these patterns are, we use a data set that's familiar to all of us and that also Adrian used in his talk just before this, where we are now projecting this pattern, which, similar to a principal component, has a weight for every gene. So we can transfer it to other data sets that also have the same genes in them. So we're looking at the strength of our in vitro pluripotency pattern in the in vivo mouse development here. And the really hot spot here is the pluripotent epiblast at E6.5 in the mouse. And as time moves in the next 2 days, I think they took either 6 or 12-hour time points here over 6.5 to 8.5. And you can sort of see it descending as you move outward toward the more committed lineages from pluripotency. And here's a map of those lineages here that are closest to the hotspot in pluripotency. And very different is the pattern of this mesendodermal induction pattern that comes on in BMP in vitro. It highlights these mesodermal lineages, and to a lesser extent, some of these endodermal lineages on the southern end of this mouse developing continent. So, I'm just going to show you a few more of these, but the time and lineage dynamics in vitro really map quite exquisitely onto very specific parts of the mouse early development. And we'll see a little bit more of that now, where we're looking at the neurectodermal patterns. So again, here, each line is doing things a little differently, but clearly this pattern 15 and 12 were the sort of front door to the neural lineage in this experiment, where pattern 15 was already on at day 2 and actually started to turn off in some cell lines by day 6, particularly in that SAO1 line that was more advanced. And then pattern 12 turns on a little later, mostly at day 4 and 6, again with SAO1 leading the way. And we can similarly map these onto that mouse embryo of single cell atlas. And here it's very different patterns than we saw in the other cases. And this sort of orange highway streaming out of the pluripotent epiblast here is the neural lineage as mapped here to the cell annotations in that mouse map. And similarly, pattern 12 is just a little bit further down that highway, leading out of pluripotency and into the presumptive brain and spinal cord tissue. So these are some of the lineage and time related patterns that we were able to dissect from this experiment. And I want to show you a little bit now how these patterns are used. So if we look at the expression pattern for POW5F1, which is OCT4, a classic maintain pluripotency gene, it's high in self-renewal, it descends in BMP induction, and it descends at a lower rate when we do SMAD inhibition. And the way the NMF does things is it breaks that down. So 2 of the 22 patterns that we asked to reduce the data set to are used to recreate that. So pattern 7 plus pattern 9, and these numbers here are how much of that pattern you need. If you simply add 4 times this pattern to 6 times this pattern, you end up with an estimate of the expression of POW5F1 across this whole experiment. So this is how these dynamic lineage-related patterns work together. But there's one other thing I haven't told you yet about the 22 patterns we decomposed from the data set. And that is, for each of the 6 lines, we found patterns that did not change across time and for the most part did not change across lineage, and were simply present in every observation of any one particular line. So this is one of those lines, which is the H9ES cell line that is widely studied across the world. And what you can see here is that all the other lines are down here around 0. So this pattern has decomposed a signal that is specific to this cell line, no matter what it's doing. Now, what does that mean? Before we go into how it sort of fits into this pattern of dynamic pieces, I want to show you how steady this is. So as I said, H9 is a cell line that's all over the world in different labs and studied all the time. So if we took this H9 pattern and we found some of this data from around the world, in fact, this is just 2 of them, but we think we have a total of 5 different experiments where we have different RNA sequencing experiments where someone did H9 as well as other lines. And if you project the H9 pattern that we saw in our experiments into those other data, H9 is always identified by them. So these are similar to the lineage bias in these cell lines that's stable across time and across experiments. The specific signature of their transcriptome is similarly stable. No matter what year or what lab these cells are run in, they're always whispering this little signal. Now, what is this signal that's flat? Most genes do not look like this. There are some, we could talk about those, but there are very few genes that are simply on in one cell line and off in 5. That's not how our genome works. We don't have trillions of genes, then everybody can pick and choose which ones. So it's really about how the same genes are used in a little bit different way. And these patterns basically represent an offset. So if we look at a different gene and see how the non-negative matrix decomposition worked, this is the OTX1 gene, and here's its pattern of expression across the 6 lines in this experiment, both conditions and days. And it is a combination of a dynamic pattern 12, that neural pattern, and a pattern that is specific to one cell line. So if you combine these 2 patterns, you see this red lifting off in certain cell lines, but then you also see just an offset where this particular line, UC06, another ES cell line, is expressing a little bit more of the OTX1 gene than the other lines. So, and this is really how it works throughout the genome. There's these very large dynamic patterns that are associated with lineage. And then there are these patterns that are very specific to individual lines, but they're used in combination. Because as we looked at the beginning, all the cell lines go to the neural ectodermal lineage or the mesendodermal lineage when pushed that way. But they move that way with different efficiencies, and as we'll see, with different regional identities. And as we'll show, it's actually we can actually connect these stable line-specific transcriptome signatures to the biases in their lineage output. So this is just to give you a scope of, these are all the 22 patterns. There's a ton of different BMP, a couple of these neuroectodermal, and then here along the bottom, there's 1, 2, 3, 4, 5, 6 patterns, 1 for each cell line. That simply is describing the genes that those lines express with just a little bit more intensity than the other lines do. Okay. So now we're going to get into the functional implications of these transcriptome patterns that are specific to individual lines. So SAO1 was the line that goes most efficiency through the neuroectodermal lineage, and IO4 is the IPS line that went most quickly toward the mesendodermal lineages. So what we're looking at here, the black line in each case is the distribution of weights for the transcriptional signature of SAO1 at the left and of I4 at the right. And the red line is a subset of genes, 250 genes that we got from reviews of the literature that look at retinoic acid response. So what you'll see is in the red line for I4, there's this extra little bump here at the right. So there's a large number of high weights for these retinoic acid signaling genes in the I4 line. And one of those genes is GBX2, we show it here. And the weight for GBX2 in SAO1's transcriptome signature is very low. So we were interested to see how this might impact functional output in a lineage assay, because GBX2, along with OTX2, happens to be sort of a team of transcription factors that defines the midbrain-hindbrain border. So Silke used a hindbrain report or a hindbrain assay. So we stimulated all 6 cell lines with retinoic acid, and the readout was HOXB1 positive cells in the dish. And here's a dose response here where you can see again that these are stable phenotypes where, and no matter what the concentration of retinoic acid is, what cell lines that are less reactive or more reactive stay that way. And what we could actually do is for all 6 cell lines, check how highly ranked those retinoic acid response genes are in that cell line's transcriptome signature specific to that line. And the number of HOXB1 cells, so these are the more posterior cells emerging in the retinoic acid response assay, the number of cells that turn on HOXB1 is directly related to how high retinoic acid response genes are in their transcriptome signature. So this means that even in pluripotency, before any induction, before any assay we do, the expression of retinoic acid genes already predict how far they will go when you actually stimulate them with retinoic acid, here read out in HOXB1. And Silke did further experiments where she looked at additional posterior neuronal fate markers to show this. And I4 versus SAO1 always has this bias where I4 is more posterior. And so what it's looking like is we can't really formally say whether it's GBX2 on its own or this whole group of retinoic acid response genes that's overexpressed in I4. But it looks like the signature is associated with the transchromic signature is related to their lineage bias in vitro. Okay, so that was the first 6 lines. We had 2 at the sort of poles that helped us understand what dimension we're moving along. And then we took another 6 lines that were derived only from 3 donors this time, where we had 2 lines from each donor. And we did a similar experiment where we had pluripotency and some lineage induction. And we did another similar decomposition with the non-negative matrix factorization. And I'm going to skip some of the lineage dynamics to say that they, for the most part, recapitulated what we saw in the first one. But what this is a picture of is the not only line-specific, but now donor-specific transcriptome signatures. So in that big mix of dynamic lineage patterns and time changing, we again found these patterns that were flat across time and flat across lineage, but specific to both lines of any individual donor. So we had 3 of those, one that matched up to each of the donors. And so there's actually 2 lines represented here at the top here, 2 here and 2 here. And the other lines are flat along the bottom. So again, we're seeing that cell lines and now multiple cell lines from the same donor actually express stable transcriptomic signatures. And at the time, this is a very cool resource because the cell lines from which, or the donors from which we derive these cell lines, we also have postmortem brain tissue. And they add also as a part of a large collection. So we had about 300 postmortem brains, 3 of which are from these 3 donors. So when we project these patterns, so look at the strength of these individual donor-specific transcriptome signatures in the brain tissue, we can, and remember, these signatures were only defined in the context of 6 lines and 3 individuals. So that would, of course, influence your power to distinguish them from others. We're distinguishing them from 3 people here, but even when we look at these transcriptomic signatures that are specific to these donors, in a large pool of 300 donors in the postmortem brain tissue, so the vertical axis now in the bottom panels is how strong this donor signature is. The one in the middle actually turns out to be the single highest value, so it's higher than all 300. The other 2 are among the top 1, 2, 3 of that. But basically what this is an indication of is that the transcriptome signatures that are specific to the lines from one donor in a dish, those transcriptome signatures are present, in this case, in the 20, 60, and 50-year-old brain tissue from the donors from which those cell lines were taken. So this is pretty exciting because if we can get functional readouts of what a transcriptome signature means in vitro, and someone's transcriptome signature is stable across a lifetime and presumably across tissues, we've seen it also in their fibroblasts, here in the brain tissue, then that opens the door to being able to understand elements of a living human's biology by studying their cells in a dish. And that's really kind of where we're going eventually. So this was exciting to us to see it in the postmortem tissue as well. Now, one sort of complication or more interesting piece to this is that there was also a transcriptome signature that was not specific to a donor, but that picked out one particular line. So this 2053-6 is actually one of the lines here. So this donor had a signature, but then this one line also had a signature. Now this line specific signature did not project into the brain. It doesn't show any enrichment in its donor. So presumably this is an epigenetically regulated pattern that arose in this -6 line at some other point, presumably in vitro, maybe reprogramming, we don't know. But what is interesting is that it behaves differently than its sister line, which is 2053-2. So in that HOXB1 retinoic acid assay, if you look at not only SA101 and I4, but now the 2053-2 and 2053-6 lines who have different transcriptomic signatures, again, their production of HOXB1 positive cells in response to retinoic acid is correlated with their expression of retinoic acid genes in pluripotency before they're even pushed to doing it. Now, in the different lines from different donors, that's going to be somehow genetically regulated. But here, in lines from the same donor, it appears it can also be regulated epigenetically. And Sung-Mei Seo in the group did some epigenetic finds. And so both globally and at individual loci, we can see that there is huge differences in the H3K9 trimethylation of genes involved in this process. So all the way at the right, this is the GBX2 gene, and you can see in the line that does not produce as much posterior output when hit with retinoic acid, the GBX2 gene has been wrapped up in H3K9, which is, of course, I'm sure as most of you know, a inhibitory mark associated with heterochromatin and reduced gene expression, but chromatin is not accessible for expression. So the expression of GBX2 in the -6 line is a result of the absence of a repressive chromatin, which allows it to more efficiently move through these posterior retinoic acid assays. Okay, so this is my last slide now to get through it. So now we studied two sets of 6 lines, and we wanted to really ask the question, how much of what we're understanding in these axes that we've drawn through small sets of 6 lines, how much of that is relevant to the wider population? So this is another bulk RNA-seq experiment from 317 lines derived from 101 different individuals from Ihor Lemishka's lab, and this was part of the NextGen consortium work. And this is donors ranked by their PC1 value simply inside this data set and colored by retinoic acid. So in any one vertical line, there are multiple lines from a donor, usually 2 or 3 lines from a donor. And as you can see, there's very little vertical variance in this, meaning that the different lines from different donors act the same in this first principal component data. And this first principal component is heavily related to retinoic acids. And to connect back to our own data, if you look at the I4 and SAO1 signature, and you look at the gene weights in this PC1, the I4 SAO1 axis aligns very well with this axis. So it looks like the axes that we found in vitro, both functionally and transcriptomically, actually live across the spread of human diversity across, in this case, 100 individuals and 300 lines. So now what we wanted to do is map this onto development. So this is the same data set we've seen, the P1 saw from Marioni and Gutkins et al. But now it's colored not by a gene or by one of our patterns from the decomposition I was showing you earlier, but by the first principal component describing variation across 300 pluripotent stem cell lines. And as you can see, low PC1, which is down here, maps to the pluripotent epiblast. And you can actually see the highway that I was talking about earlier that pattern 15 and 12 described. This sort of streaming of cells out of pluripotency to the neural lineages, but none of that is increasing PC1. We still have that purple down there. The high end of PC1 is associated with the mesendoderm up top and the mesoderm up top and the endoderm down at the bottom. So that really the PC1 here is mapping pluripotency and neural lineage on one end and the mesendodermal lineages on the other. So the natural variation across human pluripotent stem cells spreads right across the lineage map as you emerge from gastrulation, as you emerge from the pluripotent epiblast. So we wanted to look even sort of more into this PC1 and more into this variation across 300 lines. And what we found is that pattern 15, that neural pattern from before, if you compare it to this PC1, actually splits into, well, it's negatively correlated and it splits the retinoic acid genes into two classes. So what we did is actually now make this same plot that we have up here, but it's split by retinoic acid genes that are high in P15 or related to the neural lineage, or retinoic acid genes that are low in P15. So basically it's a neural and a non-neural element of these retinoic acid genes. And if you didn't notice before, in the middle of this PC1, there's a nugget of high retinoic acid. That most of the high retinoic acid is up here, but there's something in the middle. And it turns out there's a neural retinoic acid signal in the middle here. And that's what P15 helped us identify. And as we map these onto the mouse epiblast again, we can see that within those retinoic acid genes that we have, there's actually, when we split them by P15, we can see the mesendo-related retinoic acid signaling here and the neural retinoic acid signaling here. This is just a blow up of that to show that heat shows up right here in the brain. So I did run right up to 1:30 here, so I'll take questions now, but really we are excited about the ability here to map human variation onto development to understand how people are different and how pluripotent stem cell lines in vitro are different, so they can be harnessed better for therapeutic development in these models. And so I'll put these links in the Slack channel, but all of the data I showed you today, we actually have a website where you can explore all of it gene by gene. And actually I can, just to end, I can show you that. So I'll send you this link, but this is the RNA-seq data from our data set. We have the Ihor Lemishka's data and then the data from the mouse embryo all here together that you can view. You can type in any gene you want and look at it. And what we're developing just in the next couple of weeks will be actually the ability to not only look at individual genes across this space, but to project the different dimensions that we to show that. So thanks. And I'd love to take any questions if you guys can stick around past.
Yeah, thank you for the great talk. And yeah, that website and data set, thank you for sharing that. That looks great. Yeah. So does anyone
I really hope actually it can be useful to a lot of people, because there's so much data out there today, and the more eyes and hand coding we can get onto it, the better. So kind of a common place where it can all be found, because everybody has a website for their own data set, but rarely can you look at them together.
Yeah, no, that's great. So any questions? And yes, we have a chat channel where you can also ask questions, but Daniel, go ahead.
Hey, thanks for the great talk. And also thanks for doing all this, putting all your data sets online. I think like you discussed before, I think this is really the way to go for the future. We tried the same with our stuff that hopefully will come out soon. What my question was is about these patient-derived cell lines that you had. How similar would they be considered usually? So for example, if you said they were post-mortem, so assuming they were tumors, for example, would have been all the same kind? Would have been they diagnosed the same tumors, or was it cell lines where you, from the beginning would have thought they are different?
So there's no tumors involved. These are actually post-mortem cases that are acquired from the medical examiner's office. So they harvest brain, and actually it's either skin cells or scalp or actually there's also, they did some meninges, but in any case, there's, these are people who died in car accidents, and we harvested brain tissue and cells for reprogramming. I'm not quite sure I understand.
Yeah, I didn't phrase it well. So, but usually you would have thought you took the homologous cells from every patient, so to speak.
Ah, you mean the tissue source for reprogramming?
Yes, exactly. Yes.
Yes. So, actually, Soki's on the line, and you can remind me, Soki, but I thought it was from the, it's either from the dura or the scalp. Do you remember which one it was, Soki? But it's all the same for the second set of lines.
Yeah.
The first set of lines actually had 3 pluripotent stem cell lines that were reprogrammed and 3 ES. So the 3 IPS and 3 ES in the first set. The second set was 6 IPS, but all from the same tissue.
Okay, thanks.
So May is saying that the second set of cells is all from the dura. So the meninges, which actually gives you a plus that it didn't spend a lifetime of being bombarded by sunlight. But I'm not sure how big of a problem for DNA cells. But in any case, they came from the dura. Thanks, May.
If nobody else has a question, I'd really like to sneak one in, kind of piggybacking off of Daniel's question about the patients. It's 2 questions, sorry. The first one is,
sure.
Do you have information about the relationships between any of the donors? Like, do you have any parent-child pairs or sibling pairs? And we
don't. So that's a wonderful question. And I don't know if you're thinking this, but the reason it's super interesting to me is, are those transcriptome signatures genetic, right? If mom has one, does a kid have half or a quarter? Does it go with that? So we have a collaborator that has bulk RNA sequencing from relateds and also from twins. So we're beginning to ask that question, but in the data set I showed today, there's no relateds at all.
Okay, and then follow-up question is, do you have any information about, were any of the donors, did they have known diseases? Like, for example, were any of them diabetic, or a disease that might be fairly common in the population that correlates in any way with your differences that you observe?
So yeah, we have found no disease correlations. I believe there's actually 1 schizophrenic in the second set, and there's 1 Parkinson's disease in the first. But with the number of 6 and 6, we were interested in human diversity on a small scale, so we didn't really want to try to figure out anything about 1 or 2 samples. But when people have a large, the ALS community, for instance, has hundreds, if I not think thousands of lines now collected. So we will be able, not we us, but the big we will be able to ask that question as these collections of cell lines get bigger. And what we're kind of hoping to do with this work is kind of inform how to navigate those very large collections of IPS cells. How would we ask questions about them? What dimensions are they different along?
Very exciting, thank you.
One tiny other piece of your first question, although we didn't have relateds, when we projected the signatures from the in vitro to the brain tissue, what we did is we took a metric of how different the individual, because we also had SNPs on these individuals.
Okay.
So if you just take the whole genome-wide SNP profile for each one of those 300 cases we had in the brain tissue and saw how distant they were from the person whose tissue it was, there is a correlation between how genetically similar you are to that person and how strong that person's transcriptome signature is in your own group.
Wow.
So yeah, so it's kind of a way zoomed out picture of genetics there. But it's suggestive that that when we get into these relateds and things, that the transcriptome signatures are, as you might think, some kind of grand sum total that the cell is integrating across all the SNPs and CNVs that are in that person's gene, which is of course why or how all the different cells from all the lineages in a person could be whispering the same tune.
Really interesting. Thanks.
Cheers.
Yeah.
Thank you guys so much.
I think, if there aren't at the moment any questions, then we have the Slack channels for both of the speakers where people can, give their questions. And I think they're open for a week, so.
Great.
Yeah.
Thank you guys so much.
So thank you for everyone, and see you week after the next on Thursday.